So good morning, everyone. It's a great pleasure to be here with you today, and a great honor to be able to give this presentation. So you heard my name. I'm Dr. Fakhari. I'm a pediatric neurologist and neuroscientist in Boston. I work at Boston Children's Hospital and at Harvard Medical School. And I was delighted to accept this invitation by the Spastic Paraplegia Foundation and Dr. Fink, mainly because I wanted to share some lessons that we've learned over the last years, and I hope you'll find them interesting. So the title of my presentation is Childhood-onset Hereditary Spastic Paraplegia, Lessons Learned from Translational Approach to Ultra-Rare Diseases. And before I start going into our data and talk about our science, I wanted to mention that a lot what you're going to see today was made possible by small seed grants that got us started, including one from the Spastic Paraplegia Foundation. So I wanted to give a big shout out and tell you that this is actually how we start things, with grants like the ones that the Spastic Paraplegia Foundation provides. These are my disclosures. So in the next 45 minutes or so, I hope to cover 4 areas with you. The first is I'll talk a little bit about the challenges of studying rare diseases, and I do realize I'm preaching to the choir today, but I do think it's important for us to remember the framework that we operate in. In the second part, I'll talk about our clinical research, and specifically our approach to creating clinical trial readiness for childhood-onset hereditary spastic paraplegia. I'll highlight some of our work for gene and variant discovery, and I'll tell a few short vignettes about SPG3A, SPG4, SPG26, and the AP4-related forms of hereditary spastic paraplegia. Then I'll spend some time talking about our natural history study, and I think this is a topic that is very, very important for us as a society and for us as a community. And I'll highlight some of the ongoing and published work on AP4-related HSP, on SPG15 and SPG49. And then in the last part of the talk, I'll speak a little bit about emerging molecular therapies, and I'll define for you what I mean by that, and I'll try to outline a little bit of a roadmap for at least some of the childhood-onset forms of HSP. I'll speak about our program for small molecules and for gene replacement or gene therapy. And at the end, I hope we'll have a short discussion about future directions, because I think it's very important to add your perspective to all of this. So with this short intro, I'll just highlight some of the challenges that we face. And this might not be new to you, but I think it's important for us to remember rare diseases, by definition, affect only a small number of people. But if you took all rare diseases together, rare diseases affect millions of people worldwide. So I think there is a huge unmet need. Most of the 7,000 or so rare diseases are genetic, and about 2/3 of rare diseases present in childhood, and the vast majority, that is 99%, lack a specific treatment. And I think that last point is very well illustrated in this graph here, where you see plotted in the blue, with the blue line, you see the number of publications on rare diseases, and you see that that line is going up, which is great to see. At the same time, however, you see the red and the green lines, and although there is a positive trend over the last decade, that gap between the blue line and the red and the green lines is still pretty wide, and that's exactly the gap that we need to close. So how do we translate knowledge into therapies? And I think one of the reasons the graph looks the way the graph looks is because we've had remarkable advances in gene discovery. We can find genes, we can make diagnoses more rapidly than we ever could now. But at the same time, our understanding of the molecular mechanisms, so what comes after the gene, really is lacking behind, and that it's in part that's because disease models and reagents often still need to be developed. On the clinical side, we're lacking deep phenotypic studies or deep phenotyping and natural history studies. And at least historically, there's been a limited interest in funding rare disease research, both on the federal funding side, but also in the commercial space. But that's slowly changing. So I think sometimes it's helpful when you think about the future to take a look in the past. And I'll just go over this very quickly, but I think if you look at the history of hereditary spastic paraplegia, you kind of see a lot of the history of neurology there. So the first description, as many of you know, dates back to the late 19th century and the early 20th century. And at that time, clinicians in Europe, in Germany, and in France described the clinical syndrome of hereditary spastic paraplegia. They had no ideas about genetics, they had no imaging, so it was a very basic clinical syndromology. But they recognized that this was an inherited disease in many cases. Then in the 20th century, we saw pathology and imaging emerge, and the field found some common features of hereditary spastic paraplegia using these studies. And I think what has really revolutionized our understanding, or has taken it to the next level, is genetics. And that started in the mid-1990s and is still continuing. So we're up to over 80, I think at this point, probably close to 90 genes that we know can cause hereditary spastic paraplegia. And here comes the gap again. So we know the genes now, but how do we translate this into therapies? There are some emerging efforts, and I'll talk about our approach to some of them in a moment. But just to say that we're now standing on this knowledge, we're standing on the shoulders of many, many generations that have come before us and can start thinking about translating this knowledge into therapies. And we talked about this a little bit last night, but I think in my mind, as a pediatric neurologist and as someone who sees children with rare diseases every day, I think there are two fundamental challenges. The first one is we have to be visible. We have to be ready for when therapies become available to take them up and to say, here is, a cohort of patients, there is an unmet clinical need, please develop this therapy for this indication. And that takes one thing. It takes building a community. And I've listed all the other factors that we've learned need to go into this here in this little cartoon, but at the end of the day, you have to build a community that looks at all of these different things. And then mechanism-based treatments. And if you think about it conceptually, about medicine conceptually and the approaches that we use, and I'm simplifying a little bit, there aren't that many kind of therapies that we have available anyway. There are only 5 modalities if you break it down. So the first one is small molecules. So those are medications you take by mouth. That's the vast majority of medicines that we have available today. For some indications, this is not relevant to HSP necessarily, we have something called protein replacement or antibody-based treatments. And then we have 2 emerging classes of therapies. The first one is oligonucleotide therapy or antisense oligonucleotides. And this is a novel class because as you see on the right side of the slide, this is a class of therapeutics that works on a different level. It works on the RNA level. So we're getting to the root of the problem. The same is true for gene replacement, which is working at the DNA level. So we're moving from what's the downstream pathway to what's the most proximal target. And I think that's very exciting. So I get asked a lot, what do I mean by clinical trial readiness and what are the initial steps? So we put this on paper here, and I think this could be sort of a rough draft of what this could look like for HSP. I think the first is you have to have a broad and rapid gene discovery platform or genetic diagnostics platform. You have to be able to make a diagnosis quickly. And there is not a single week that I don't learn of a very long journey that a family had to go through until they finally received a genetic diagnosis. And I think we have to do better. I think we have to shorten that time. So the goal here is an early diagnosis and a molecular classification. The second step is we have to create an international patient registry and biorepository. And I emphasize the word international because that's what we've learned. In rare diseases, you have to work across the globe. It's just impossible to do this in the United States alone. It's impossible to do this in any country alone. So we have to work on an international level. And that serves the goal of community building, visibility, and access to care and advice. The third key element, which I'll detail in the next couple of slides, is a natural history study. And here we need to define clinical outcome parameters. We need to involve the patient community and look at patient-reported outcomes. And that's what the FDA cares about more and more. What matters to the patient? Does it matter what we measure in our clinic, or is it something that you would measure at home with a wearable device or with other avenues? And this is a term that is well known to you, I'm sure. We have to find a biomarker. A biomarker would make our clinical trial design much easier because we would have a surrogate that we can objectively measure. So we have to look at all of this and develop a plan and a very rational approach to designing a natural history study. And then the last point, I think, to me is the one that is the least clear, which is we have to come up with innovative designs for clinical trials in rare diseases because we just don't have the numbers. We can't do a clinical trial with 1,500 participants. We will do small clinical trials, and we have to be mindful and smart about how to design these trials so we can really take lessons away from them. Okay, so with this intro, I'll now go into the specific points. I'll first tell you a little bit about why I think rapid genetic diagnoses are important. I've already mentioned that this is a diagnostic odyssey for many patients. That's number one. And number two, I think we'll move away from thinking about the symptoms as the main determining factor for which therapy to choose to a classification where we use the specific molecular variant as the target for any therapy. So it's very, very important for us to have a broad genetic discovery platform because we can triage specific mutations to a specific modality. And that will become more clear in the second half of the talk. I think if this was a big enough approach, then we would also learn about more complex variants and we would start learning about risk and trait-associated alleles. This is not my area of expertise, but if you have thousands of information and genetic information from thousands of people, you can not just only pick up mutations that we know are associated with disease, but you can understand risk and genetic risk scores better. And then one or two aspects that I think are very relevant to HSP are, one, there's clinical heterogeneity. That means that one symptom or one phenotype, as we call it, can be caused by many genes. And I think HSP is the prime example for that. But we also see that there is something called phenotypic pleiotropy. And what that means is that one gene can cause many symptoms. And the more we sequence, the more we find gene mutations, the more we learn that the symptoms are actually much broader than we initially thought. Okay, so how does this look like in practice? So when I see a child with a syndrome that involves progressive lower limb spasticity, and in many cases other symptoms, we very quickly go to molecular testing. And we know that the yield for molecular testing is pretty high in this situation. But you have to remember, and this is just a very quick educational section here, you have to remember not all genetic tests are the same. So there are gene panels, many will be familiar with that, but not all panels are the same. And we have something that is a little bit broader. It's called exome sequencing, which looks at all the coding regions of the genome. And that test is very good. It's sort of our first-line test now, but it also has limitations. There are also mutations that we can't pick up with exome sequencing. And then there is the next level of resolution, if you will, and that's genome sequencing. And we have that available on a research basis and increasingly also on a clinical basis. And all of that is very important because we need to know exactly what the mutations are so we can design therapies against these mutations. Okay, so in our practice, we usually go to exome sequencing for the reasons that I stated. But we also realize, and this is probably something unique to where I practice, we realize that we see a lot of children who have hereditary spastic paraplegia, the clinical syndrome of hereditary spastic paraplegia, but have a negative exome. And that can have many, many reasons, one being the limitations of exome sequencing. So we decided to launch a study where we are able to offer to families that have had clinical testing a next set of tests on a research basis, and that's whole genome sequencing and other modalities. And we've just launched a study, as you can see here, in April. And so far, we've enrolled 17 families. So we're hoping to be able to provide a diagnosis to these 17 families using this additional research test. So I just want to put that out there as another frontier, I think, in genetic discovery. And we can do this because we build on something that our institutions have created, which is called the Children's Rare Disease Cohort. And we build on this vast infrastructure that allows us to do this. And not through this study, but through our natural history studies, we've learned a couple of lessons about genetics and HSP, and I want to share them briefly. So as you all know, there are forms of HSP that are passed on from generation to generation, and many of these forms are what we call autosomal dominant forms. I'm listing the most common ones that we see here. And in this group, there's a unique challenge for us in that we see children where the mutation occurred for the first time. We call it de novo variants, or de novo mutations. So I'll spend a little bit of time talking about these specific cases in a moment. The bulk of what we see in terms of genetic findings falls in the category of autosomal recessive forms of HSP. And I'm listing the most common ones that we see here. And there are other challenges. There are other challenges including certain modes of inheritance and novel missense variants. Then we are seeing patients where we don't know what the gene is. So these are genes that still remain to be discovered, and that's a huge challenge. And the last point, I'm not going to go into much detail, but it's also for us to remember, and this is more for the clinicians in the audience, that there are conditions that look like HSP but aren't HSP, and some of these are treatable with other types of therapies. That some of them are inborn errors of metabolism, and we can actually treat them with a specific diet or a specific medication. So it's important to remember that as clinicians. So now I want to highlight the first point a little bit and speak about the de novo cases, because that's something unique in our populations of children with HSP. So we were faced with a scenario where we saw 5 children with rare forms or rare variants, de novo variants, in the ATL1 gene for SPG3A. And these patients had very, very different symptoms. They had much, much more severe symptoms than you would expect. They had, as you can see on the top left, a very early and severe involvement of all 4 extremities. And they had problems with speaking and problems with swallowing. And they had problems, or they had findings on their brain MRI scans, as you can see on the top right. And although this was just a small cohort, for us it triggered a critical review and critical thinking of all the variants in ATL1. And you see them mapped at the bottom. These are all the published variants, and highlighted in red are all the variants we discovered. And at the top, you see the de novo variants. At the bottom, you see the non-de novo variants. And this is a collaboration with Dr. Blackstone and others. And a very talented student in my group, Julian Aleko, he very carefully looked at these mutations. And we have tools available now that can help us, one, determine statistically if a variant is associated with a specific set of symptoms. And that's what he did here. He essentially found that de novo variants present earlier, and he found that they lead to more severe symptoms. But taking this beyond just description of the symptoms, we also used very powerful prediction tools and 3D modeling of the protein structure to look at where these mutations are located. And we found that they're all located in the same neighborhood. And not just in the de novo cases. Interesting, when Julian looked at all published familial cases, and specifically those cases where the symptoms would go beyond what you typically see in SPG3A, he found that these mutations were also in the same neighborhood. So that teaches us something about the biology. And I'm not an ATL1 expert like Dr. Blackstone, for example, but the elastin expert, but the lesson here is that rare presentations on the extreme of the spectrum can tell us something about the biology. I think that's the lesson that I'm taking away from this set of analyses, and that's summarized here, basically. So that was one example. And around the same time, we also noticed something very similar in a cohort of children with SPG4, with SPAS mutations. Again, all presenting with de novo mutations. You see some of the characteristics of the cohort listed here, all presenting very early with symptoms in infancy or the first 2 years of life. And again, this was a large collaborative effort with many centers around the United States and in Europe. And you see at the bottom listed the mutations. And I think you don't have to be a geneticist to recognize that most of the mutations are on the right side of the slide, they're in the right corner. And they're actually all except one in the AAA domain of this protein. So again, that teaches us something about the biology and how de novo variants might lead to a different clinical presentation. That's essentially summarized here. So a phenotype or symptoms that are very different from what you would expect with SPG4. So symptoms that start in infancy, prominent developmental delay, and progression over time if you look at a cross-sectional analysis. So a couple of lessons. Again, all of these children and the brave families that participate in this research, they teach us something. And the lessons we take away from this first initial analysis, and this is just coming together and thinking about it, is that children with de novo variants in genes classically associated with what we call pure forms of HSP might present with complex features. Mutational clustering, as I've shown you for Atlasted, can help us identify genotype-phenotype correlations. That's very important. Phenotypic differences and mutation clusters potentially suggest diverging molecular mechanisms. So not all mutations might do the same. It's possible that some mutations in one neighborhood do something completely different than mutations in another. And for me, as a pediatric neurologist, it's very important that clinicians are aware of this because inevitably, many of these children will be misdiagnosed and will not be referred to genetic testing because the symptoms are so different than what is in the textbooks. And I think it also is a call to arms because we need to understand the molecular mechanisms better because that has implications for therapy development. Okay, so that was a quick vignette on what we learned from children with de novo mutations and autosomal dominant forms of HSP. And now I want to speak a little bit about the autosomal recessive forms and specific situations here. So again, by the way of where I practice, we see a lot of second opinions. We see a lot of unclear cases. And I just want to illustrate one case of a young patient of mine here because I think it illustrates why we need to think and go the extra mile in these cases. So this is a 13-year-old girl with a history of developmental delay early on, and then over the years, some cognitive decline, and a progressive picture of spastic diplegia with gait abnormalities, and also a peripheral neuropathy, and she had no unifying diagnosis. As you see listed here, she became symptomatic early in childhood, but then there was this progressive picture, and specifically in the 1 to 2 years prior to coming to Boston to see us, she had a relatively rapid decline in her motor function. Her brain and spinal cord MRI were normal, and she had genetic testing. She had an exome, clinical exome that was negative. Now, when this is a short video just showing her gait, so a complex picture, but the point I'm trying to make here is in these cases, one should never stop looking. And what we did is something very simple. We reanalyzed the existing exome data. We used a different filter set with knowledge from 2022, not knowledge from 2019. And that led to the discovery of 2 variants in a gene associated with hereditary spastic paraplegia type 26. And importantly, we realized that this gene is coding for a protein that is an enzyme. And when you have an enzyme, it has a certain function. It has a substrate and a product. So it gives you an opportunity to really test the effect of these mutations. So Again, this goes into the very sort of fine details of genetics, but if you look at the mutations, the second mutation, the one on the right, is a missense variant. And we're often not sure about those because they're a small change in the DNA and we're not sure if they have an impact. But again, this is an enzyme. So my team looked at this and we realized this is an enzyme in a very important pathway, one that produces a certain type of lipids called gangliosides. And you see in red the enzyme listed here. It has all of these different functions. So it became a testable target. And my team reached out to collaborators in Japan who discovered this enzyme. And we asked them, would you be willing to look at these mutations that this girl is carrying to see if they lead to any impact on the enzyme's function? And they said, yes, of course. This is why we study this enzyme. So we teamed up with the lab in Japan, and as you can see in these graphs here, there is a peak missing, a peak for GM2 and a peak for ACLO-GM2, and that proves that these two mutations lead to a loss of function. So I'm highlighting this case here not because I think it's relevant for thousands of people, but it was relevant for this family, and I think we can do this in the right hands and with collaboration. Now, for the AP4-related forms of HSP, we came up with something very similar, what we call a functional assay. So an assay in a cell that can tell us if there's a loss of function or no loss of function. And that's hugely helpful, especially in the situation where you have novel variants. And in fact, this test is a test we can do in 2 weeks in the lab. It's a relatively cheap test, and it has pretty good sensitivity and specificity. So in these situations where we're not sure about the genetic diagnosis, we can employ this test and we can confirm a mutation that way. So I think it's a very important tool, and we need more of these tools. Okay, so now I'm coming to the natural history study part. And, oh, thank you. Oh, sorry about that. Okay. Here we go. Thank you very much. So now I'm coming to a more clinical part, and I think this is relevant for all of us. So when we first started this program for rare forms of childhood-onset HSP, we identified a natural history study as one of the priorities. And it took us a while to launch this, but it's now up and running. And we specifically focus on the pediatric population. So these are individuals who have symptoms of HSP or molecular diagnosis of HSP with symptom onset before the age of 18 years. And we're only really picking up pace with this because COVID slowed us down a little bit, but we are quickly recruiting, and I hope this will be a platform for many of the forms of HSP that present early on. So we conduct what is called an initial study visit, where we go through questionnaires and rating scales. They're listed here. We're collecting MRI scans. We're collecting blood samples for biomarker discovery. And then we follow up every 6 to 12 months with a follow-up visit. We go through a shorter questionnaire. And this is all designed to detect change, because ultimately what we need to find are things that change over time, because that will be our clinical endpoint. That will be our natural our historic control. So thanks to many of you and many other families, we've been able to enroll 360 patients so far. And you see the distribution listed here. We started this specifically for the AP4-related forms of HSP, and we've since opened it up to all childhood-onset forms. But I think the numbers are still reflecting that we have a specific focus on the AP4-related forms. But you see that many of the early onset HSPs are already represented. And families joined the study from far. 35% of the participants are in North America, but the rest is coming from around the world. So it's really remarkable to see the patient community come together that way. And we are enrolling on a rolling basis, and we're conducting the second and third study visits. And at this time, we're up to 80 second visits, and that number is expected to increase. There is a drop-off rate, so there are families who participate for the first visit and whom we cannot meet for a second or third visit. That's expected, but I expect these numbers to slowly increase. So as just a vignette for this natural history study part, I just want to introduce the AP4-related forms of hereditary spastic paraplegia. So the AP4 stands for adaptive protein complex 4. It's a complex with 4 subunits, and these 4 subunits are kind of like the 4 wheels of a car. If you're missing a wheel, you can't drive. So it really doesn't matter which one you're missing, it all leads to the same problem. So we think of it as one disease, really. And just to illustrate that point, I'm showing you 3 videos here of patients of mine, and you can see at the bottom that they have different mutations, including in different subunits, but they present with very similar problems. So with early developmental delay, with initially low muscle tone, and then an evolving spasticity in the legs. So we think of this as one syndrome, although it's 4 different genes. And we started the registry natural history study with 2 aims. One, we wanted to establish an international registry and perform a first cross-sectional analysis. I should have mentioned that when I started working on this, there had been a dozen patients reported worldwide. So it's an ultra-rare disease. And we first wanted to learn about the spectrum of symptoms. So we designed a registry and designed a cross-sectional analysis that was very comprehensive because we didn't want to miss any symptoms. So that was the first objective. And the second objective was then to launch into a longitudinal study. And I won't have time to show you all of the data that we were able to capture for the first aim, but the publications are listed here. But I want to highlight a couple of specific things. So the first thing that we learned is that the first symptom is not spasticity. The first symptom is global developmental delay and very prominent motor delay. So you see the motor milestones listed on the left. You also see the age of onset summarized at the top right. And you realize that symptoms start in the first year of life. So they start very early. And you also see the age at a molecular diagnosis, so when genetic testing was confirmatory. And the average age in this initial cohort was 10. Now, these are patients that live in different parts of the world, and access to genetic testing is different based on where you live. But still, that number is astonishing. It's bothering me because it takes 9 years for us to make a diagnosis where it should really take 9 months at most. That's lesson number 1. Lesson number 2 is spasticity becomes the predominant feature, the predominant symptom, and we see a pattern of progression if you look at age, if you look at the cohort and age in a cross-sectional way. And that's also reflected in some of the scores that we apply. Now, I will say that there's a little bit of a caveat here, because for childhood-onset HSP, or for HSP in general, our clinical scores aren't perfect. So we have rating scales where we determine how severe a symptom is, but none of them are perfect. And I think we, as a field, have to tweak them constantly to be able to detect changes as fast and reliable as we can. Now, one other important aspect, and I think this is also important for therapy development, is to look at health-related quality of life. So what matters to families? And we do this using a methodology that was developed for the cerebral palsy research field, and use a questionnaire that looks at health-related quality of life. And we found that the degree of motor symptoms, so that's spasticity and weakness, is the main predictor of quality of life. Interestingly, it is the degree of intellectual disability or the occurrence of seizures do not seem to impact health-related quality of life the same way the motor symptoms do. And then we took a very systematic approach to looking at brain MRI scans, and we found not only a diagnostic signature, but we also found a lot of things that teach us about the biology of this gene. So in a way, these MRI scans are a window into what is happening when this gene is missing in brain development. So we noticed a thin corpus callosum was very common, a thin white matter in the periventricular region was quite common, and we also noticed something that hadn't been appreciated and is clinically important. which is that there is a higher prevalence of brain malformations, including some that are very subtle, such as polymicrogyria. And that's important because these can produce seizures, and we treat the seizures differently if we know this is present. But conceptually, all of this work, and again, we started with 12 patients, and now we're up to just over 200 patients for AP4. We learned that this is probably both a neurodevelopmental and a degenerative disorder. So there are features that are progressive, and there are features that I think are developmental. And that, again, is important if we think about things like a therapeutic window. When do you have to intervene to really have a big impact? And I think this is a lesson for many early onset HSPs. We have to develop an understanding of when symptoms start and how they change. Okay, so another example I want to highlight here is SPG15, and this is a research study that was made possible through many families with SPG15 who came to see us in Boston, and we collected information and in a systematic way along the lines of the natural history study that we run. And you see the mutations listed on the right, and you see 2 videos on the left, just to illustrate the spectrum of symptoms. So there's a wide spectrum of symptom severity here. And I'm raising this point because, of course, our tools need to be sensitive enough to pick up changes both at the mild and at the severe end of the spectrum. Now, we applied the same systematic approach that we applied to AP4 to SPG15, and we looked at brain MRI scans, identified a diagnostic signature, also identified correlations between certain MRI metrics and clinical outcomes. And this is just emerging. The numbers are small, but I think this sets a precedence for doing something similar for many forms of HSP. And importantly, when we looked at an emerging blood biomarker, now this is a protein called neurofilament light chain, something that my colleagues in adult neurology measure in disorders like Parkinson's disease, Alzheimer's disease, multiple sclerosis. We looked at this in SPG15, and we found actually that the levels are increased in blood. Now, this is one measurement, so we don't know if they're changing over time. But if you took age-matched controls, and if you took children and young adults with SPG15, you see a significant difference. So this is important because it teaches us that this set of markers could be potentially a longitudinal marker that we can follow, but that remains to be shown. And the same is true for some of the rating scales that we use. So here you see summarized the spastic paraplegia rating scale scores, you see the SPATAX disability scores, and the point here is that there is a trend. And that's important for us to see because that means we're on the right track with the tools that we're using. They're not perfect, but we're on the right track. And importantly, and this is the effort of many families who participated in this specific study, we were able to map out the first symptoms. So we looked back, we did a very structured review of very early symptoms, and we discovered that symptoms started long before the spasticity became apparent. It was something that didn't reach a level where clinicians were concerned enough to think about HSP, but there were subtle symptoms long before the spasticity became apparent, and that's an important lesson, I think. And I think if we look carefully, we'll find this in many other forms of HSP. And then just to make that last point a little bit more clear, we compared our findings to the published literature. Now, the published literature, of course, is a mix. Gene discovery studies, clinical studies, patients who are in the adult range, patients who are in the pediatric range. But the point I'm making here is we detected a lot more developmental symptoms. We detected a lot more early symptoms than were reported in the literature. And I think it's not that there is a difference in these cohorts. I think it's just how carefully you look that makes a difference. Okay, so with this point, I'm switching gears again and talk just very briefly about SPG49. So this is a rare form of childhood-onset HSP, and we got together with an international consortium and looked at the symptoms. And the numbers are small here. You see these were 25 patients. But we realized that there were many other symptoms other than spasticity that predominated in this disease. And That's important because it led us to expand the phenotype and reclassify this disorder as a form of hereditary sensory and autonomic neuropathy. So again, this is just a value that we're adding with detailed phenotyping. We're able to provide a better classification. Okay, so with these examples, I'll now move into the part where I'll talk about therapies. And I'm coming back to the summary slide here just to highlight three classes of therapeutic modalities: small molecules, antisense oligonucleotides, and gene therapy. And I tried to map this out a little bit and come back to a thought that I shared early on. I think in the future, it will matter less what symptoms you have. It will matter more what kind of mutation you have, because the new treatment modalities like antisense oligonucleotides or gene therapy, they work for a specific kind of mutation. And it's almost secondary which gene that mutation is affecting. It's the type of mutation that counts. And I've listed here, to the best of my ability, and I'm not an expert in developing all of these modalities, but I'm listing here, to the best of my abilities, the different approaches for different mutations. So if you have a loss of function mutation, as is the case in, for example, the AP4-related forms of HSP, gene replacement is a good strategy. It's the most proximal way of fixing the problem. If you have a mutation that is exerting what we call a dominant negative effect, so this is a mutation on one copy of the gene, on one allele, that is going awry, that is disrupting the normal function of the protein, then you have to block that mutation. So it's the polar opposite to replacing the gene. So you have to have that understanding, and this gets very technical, but just know that based on what mutation you have, there are different approaches one could pursue. Now, one that we focused on that would work for many different mutations would be a small molecule. And if you look at all approved therapies, you realize that the small molecules are still the main class of medications that are brought to a clinical stage. And it is astonishing, however, to remember that of the 3,000 or more disease-associated proteins that are well characterized, less than 700 are actually targetable with a small molecule right now. So there is a large margin and a large proportion of proteins that we know are disease-associated for which we have no molecule yet. So there is still a lot of potential to find molecules, and high-throughput screens, which is typically how these molecules are identified, are ever evolving. And at least in theory, drug repurposing is possible. So a medication that may have been developed for cancer or for a different indication could have relevance to neurological disorders. You just have to test it. And I know I'm running short on time, but I just want to highlight one aspect of our small molecule work, which is looking again at the adaptive protein complex 4. And I'm going to summarize 4 years of work in 3 slides. First, we and others learned that the AP4 has an important role in the cell, and it has different proteins downstream of it that rely on its function in the cell. We identified those set of proteins. Collaborators in other labs found the same proteins. They're ATG9A, DAGLB, SERING1, and SERING3. Now, why is this important? It gives us a tool. It gives us a tool that we can look at. So I think anyone without any background can tell that the left image and the right image, they look different. On the left, you have this green signal, this intense signal in certain parts of the cell. And on the right, you have a lower signal, and it's more evenly distributed. And when you say something like that, it doesn't explain the disease, but it gives you a surrogate. It gives you a tool. So we can now ask, what intervention makes the cell on the left, which is from a patient with SPG50, look like the cell on the right, which is the same type of cell taken from his father, who has no symptoms. So that's essentially the approach that we take. We developed a high throughput platform to test this, very much like what pharmaceutical companies do. And we use this platform basically to screen small molecules We also are conducting functional genomic screens, so using CRISPR to get at the molecular target. And there's various orthogonal strategies that are looking to confirm what we find in the first two approaches. So showing you some work on the drug discovery end, we first conducted what is called a drug repurposing screen. So we took 4 libraries with compounds, most of them FDA approved, and we used exactly the assay that I showed you. We asked which ones of these compounds are making the cells on the left look like the cells on the right. And we measured this in millions of cells in each run. And we found out of these 3,000 compounds that are mostly in clinical use or have been developed for clinical use, we identified 36 active compounds. So that was the first sort of lead. But these are only known compounds, and there's sort of the off chance that we'll find a compound that is already in clinical use that we can repurpose. We also wanted to do this more broadly. So we teamed up with a pharmaceutical company. They liked our assay because it's such a robust and reliable assay. And they asked us to screen their library of 30,000 compounds. So we're a magnitude higher now. And we did this, and my team did this in 9 months. It was quite remarkable. And we identified 442 active compounds. And this was, at this point, 2.5 years ago. And we've since then worked our way through confirming and confirming, crossing off compounds off the list that didn't perform well in orthogonal assays, so additional tests. And on the drug repurposing end, we landed on one class of compounds that work in all cellular assays, including in neurons derived from patients. And on the pharmaceutical companies' library side, we landed on 3 compounds that work in all these stages. Now, you have to remember that all of this is still testing in a Petri dish. So we're looking at cells, and this is the early stage of development. But we're hoping to create a platform where we can take these molecules now into testing in vivo. So that means into zebrafish and mouse models. And hopefully they will prevail. Hopefully they will change the phenotype, and they will be something that we can develop into the clinic. So that's just an illustration of how you develop small molecule drugs. And my team knows this. I'm a very realistic person. I think about this systematically, and this is something that is up in my office, which is sort of the key steps in this program. If we assume that we have a success rate of 80% at every step, if you map this out, we land at a success rate of 13% at the end. And this is optimistic. So we're very realistic about this being a long shot, but I think it's still worth pursuing because there is a chance that we'll find something. Now, for the last few minutes, I want to switch gears and talk about a different modality, which is called gene therapy. I think you've all probably heard a lot about that already. And I thought it would be helpful for this audience to map out 2 things. One, what a typical workflow looks like for a gene therapy. And 2, what the limitations are for gene therapies for brain disorders. So the first part, and this is experience that we've made with developing gene therapies with 2 teams, one in Texas and one in the UK, for SPG47 and SPG50. You first have to design a therapeutic entity, and that's typically an AAV vector. For brain disorders, nowadays it's often AAV9. You have to decide what the route of administration is, and you have to decide this early on because all your animal studies will use the same route that you propose for your clinical trials. It's an important decision, and there you see 5 different routes listed here, and what that tells you is that the fact that there are so many listed here that there is no gold standard. They're all being pursued, and they all have limitations, and they all have advantages. The next step is, and this is something that we focus on in the academic setting, is to do what we call proof of concept. So if we give this gene therapy to a cell or a mouse, does it lead to something good? That's the proof of concept stage. And all of this, these first 3 steps, we can do in an academic setting. We can do at a university. We can do at a lab, at a hospital. But then you have to bring in other partners, and that concerns manufacturing, which is the bulk cost in all of this is the manufacturing. You have to manufacture the vector in a clinical grade, and that's not trivial. And then you have to perform safety studies. You have to perform toxicology studies, and the FDA will usually require a two-species testing. In the case of AAV9, it's often 3 species because they require testing in non-human primates. So now that you have an AAV9 vector that has gone through proof of concept testing and has shown an acceptable safety profile in toxicology testing, there are still some questions that are unanswered, and these are the questions that we're wrestling with. So what's the optimal gene dosage? What's the optimal dose? I think no one can give you a firm answer on this. I think we as a field have to optimize the expression in target cells because we want this therapy to go to the cells that are affected. We don't necessarily want this therapy in all cells of the body. And then I think the holy grail there is, can we regulate expression? Can we find a clever way how we can turn a gene off or on in sort of in a regulatable way? Delivery to the CNS is sort of where I have the most concerns. Are therapies actually getting to where they need to go? Are they, many aren't crossing the blood-brain barrier, so we have to use alternative routes, which means a lumbar intrathecal application or a cisterna magna injection. How specific are they for certain cell types? I think that's also a very important question. Now, in general, but even more so in the pediatric forms, what's our window of opportunity? When do we have to intervene to have the greatest impact? And I think that's something that we have to think about very carefully. How do we measure that the therapy is working? Of course, the first studies will all be about safety, but we want to learn and know if it's working because we're investing so much time and resources into this. How do we handle toxicity? How do we devise an immunosuppression regimen that is safe and tolerated? I think these are big questions. So all of these questions, everyone in the field is struggling with. Everyone is thinking about, and I think we'll have answers to all of them. It will just take a lot of time. And what I'm really hopeful about, what I'm really grateful for, is one, that there are many groups who are pursuing these kind of therapies for many disorders. So we will learn from each other. And I'm listing one initiative here, which is called the Bespoke Gene Therapy Consortium, which is an NIH initiative. And the main mission of this consortium is to develop a platform approach to gene therapies. A platform approach means that we're using the same technology, and we're developing a platform where this will become plug and play. So you have the same technology, and for each disease, you just exchange the gene. And I think there's a lot of merit in this approach because, one, we will systematically learn about the pros and cons of different technologies, and two, it will eventually de-risk the process and it will bring down cost. So I'm really, really, really hopeful for these kind of initiatives. And we nominated SPG 47 and SPG 50 for this initiative, and we're very grateful that these two disorders were chosen among the 14 for this first round of potential funding. So we now have an opportunity to submit a proposal and hopefully be one of the 5 or 6 disorders that will make it in the first round of this initiative, which I hope will open the doors for many HSPs. So the point here is we're trying to put HSP on the map, and maybe we're successful with one of them, and that will open the doors for others. Okay, so I'm almost at the end. I just want to sort of summarize and say, I think there are unique challenges for us in working on rare diseases and dealing with rare diseases on a day-to-day basis. But I think there are also unique opportunities. I think it's important that we have broad access to genetic testing and that we really invest in long-term invest in natural history studies because they will be very important. I think that molecular mechanism-based therapies at today's age include several modalities, and there might be others in the future on the horizon. I think that understanding the molecular mechanisms will allow us to select the right therapy for the right mutation and the right disease. And I think understanding the molecular biology or cell biology, as I've shown for AP4-HSP, also provides the opportunity for unbiased phenotypic screens, where you don't have a hypothesis, but you basically just ask, of these 30,000 compounds, are there any that could help? And then I hope I was able to show that gene therapy or gene replacement strategies for brain disorders is an evolving field. And I think we will learn a lot in the near future about this approach for brain disorders. So with this, I'm actually at the end, and I have to thank the many, many people that are involved in this work. So it's my group. You see on the left, it's a young and very motivated group. And then I have to thank the many, many collaborators that make this work possible. So we take a very collaborative approach to all of this. We collaborate with people around the world because we know that's the only way to push rare disease research forward. And as you can see listed at the bottom, we also had funding that allowed us to do all of this work. And I think we're on a good track with many of these programs. If you're interested in participating in any of these studies, or if you know people who would like to participate in this study, please reach out to us. We're always available. Catherine is our lead research coordinator, and she's handling both studies, so please feel free to reach out to her. So with this, I'm actually at the end, and I thank you for your attention. Thank you for inviting me to speak here today, and I look forward to your questions. Sure, I'd be happy to take any questions now or later. Yes, please.
Hi, thank you very much. That was really very interesting. I'm actually a nurse case manager, and I case manage NICUs all the way through to the adult world. And one of the things that has truly struck me with this and looking at the age in your area of expertise, first of all, the pediatric community has a much more collaborative feel. Always, when I deal with things, and we're a level 3-4 NICU, so what we see and the pulling in of geneticists and things like that when we see a newborn or preemie that's has concerns. So I know, like right at that level, there's a growing recognition of needing early diagnosis. Yeah. But having a daughter who has struggled through that childhood piece, how to again, increase the level of knowledge at that pediatric level so that earlier diagnosis can be captured, so that obviously more assistance and treatment may be option. But I also see a lot that happens that when people start to age out of the pediatric world and go into the adult world, there's kind of this big drop-off that you are now in with your basic neurologist or whoever, who really is not as informed because this is considered a pediatric start point, and how to do a better job of translating what you're doing, treatment options, and some of this, and recognizing that somebody may not get maybe to the point of being in the adult world to get a diagnosis, but really it's the pediatric world who's doing that piece. And how do we get that translation to happen so somebody can get an answer sooner and have more information, and then also be able to translate that to the next generation or their broader family, that should they all be looking or doing some kind of testing? I just wonder if you have any thoughts, or is there a way to spread this so that people don't fall through that, okay, now you're 21, sorry, you're done. And I know that cardiology, for example, is a group that's also now has a whole bridge group of cardiac anomalies in childhood, but now we need cardiologists in the adult world to
Yeah.
know how to hand it off to.
Yeah, I think you raised some very, very important points and a wonderful perspective. These are exactly the things we struggle with on a day-to-day basis. So I'll try to sort of answer the different aspects in order. I think in terms of raising awareness on the pediatric side, I think we've come a long way, but there's still a lot of work to do. And I think when I think about sort of what are the most common diagnoses that were given before we made a genetic diagnosis of HSP, I think by far the most common diagnosis is cerebral palsy. And I think the cerebral palsy field, or my colleagues in that field, are doing a wonderful job recognizing that many of the genetic disorders that in early childhood might look like cerebral palsy can be detected earlier if you use genetic testing earlier. And I think so there has to be a push on that end to introduce genetic testing early on. Now, how we practice across the United States is very different. In our clinic, any child who would present with a constellation of symptoms that would suggest a genetic cause would have genetic testing. But that's not true across all hospitals in this country. So I think that's number one. On the adult side, I do think there has to be more awareness, and I do think there are patients who, on the adult side, who wait for a genetic testing or genetic diagnosis for way too long. So I think one way to approach this, and also to approach your question about transition of care, if I understood that correctly, I think that's exactly the gap that you're describing. We struggle with this every day. When one of my patients, when they are in their 20s, we have to look to see if we can find an adult neurologist who's comfortable managing their symptoms, who knows about HSP, who keeps in touch with us, keeps in touch with research. And I think a way to solve the problem of awareness and to solve the problem of transition of care is to build centers of excellence for HSP, where you say, okay, here you have to have it be designated a center, you have to have a team that understands HSP, that has resources, the appropriate resources to care for patients with HSP, and it also enables transition of care. So I think with that, we will hopefully be able to form teams that can take care of children and adults with HSP across the age span. That would be my suggestion anyway.
All right, guys, we're going to have to move on, but I want to remind you that later this afternoon, we're going to have a program with the doctors. We're going to have a question and answer with them.
Yes.
We're going to have all four of them up here today. So we have to